Hello, everyone. Welcome to today's webinar. I'm Melissa Romero, Forensic Science Consultant and CNN Media Group. I will be moderating today's event, The Potential of NAMs, Fulfilling Critical Unmet Drug Discovery Needs. This educational webinar is sponsored by Eurofins Discovery. Eurofins Discovery built its industry-leading reputation on successfully delivering best-in-class drug discovery services to any organization, large or small. No matter where you are in the drug discovery and development process, or if you need full end-to-end solutions, their experts support you every step of the way with purpose, precision, and innovation. As a global leader in drug discovery for over 40 years, they have strategically amassed a deep and broad range of over 12,000 assays for every type of research, as well as a full team of industry experts across disciplines to collaborate with you. CNN works with sponsors to identify topics of interest and value to CNN's audience and consistent with CNN's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look at the help widget at the top right corner of the screen or type your query into the Q&A box. If you were disconnected during the webcast at any time, please log in again according to the instructions you had received earlier. You're encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, and as your moderator, I will be posing as many as time permits. This webcast allows certification of participation. To receive your certificate, please click on the certificate icon below your Q&A box to download your copy after watching the webinar. Please note that CNN does not endorse any company, products, or services that may be mentioned in the webinars, and each webinar will be archived at CNN Online after the live webcast. During the presentation, we will be hearing from Dr. Alistair King. Dr. Alistair King is head of biology at Eurofins Discovery. Alistair has over 25 years of experience with all stages of drug discovery from target validation to IND package generation, largely for oncology and inflammation. He has experience leading international strategic initiatives with drug discovery teams, for example, that which discovered Taflinar, and the coordination of due diligence studies on external assets for in-licensing and subsequent clinical development, for example, McKinnis. Now let's get started. I will turn it over to Dr. Alistair King.
Thank you, Melissa, for that very kind introduction. My name is Alistair King, and I am here today to speak to you on the subject of NAMs and their critical role that they're playing in fulfilling unmet drug discovery needs. What I'm going to give to you today is an overview of various aspects of NAMs, starting with what they are and their applications in drug discovery. But by no means is this a comprehensive review, more to really give a flavor of some of the things that are going on in the NAMs field. So what we're going to cover today is we'll look at NAMs, or novel alternative methods, and why they are needed. What exactly are they, and why are they needed for drug discovery, and where do they really fit into drug discovery? We'll then talk about some of the biological models that are effectively NAMs in their own right. They're replacing or have the potential to replace animal models, and really that drive for greater physiological relevance in those models. And when you think about what a NAM is, a novel alternative method, it's an alternative to those in vivo models that get closer to the human system. We'll then take a look at some of the existing platforms that are available, in some cases platforms that have been used for many years, and how they can be optimized and have been optimized so that their performance now is a lot more representative of the way drugs act in a clinical scenario. Of course, everybody has heard about the impact of artificial intelligence, or AI. So we'll actually talk a little bit about the role of AI in the context of NAMs and how that can potentially either dovetail with NAM approaches or in its own right become a NAM. And then we'll talk about, lastly, the potential leveraging of bench to bedside and then bedside back to the bench again. Are there clinical data out there that we could potentially use in this scenario? And finally, I'll summarize with some of the learning points that we will have discussed today. So what are NAMs, and why really are they being promoted? As I mentioned, NAMs is an acronym, and it stands for Novel Approach Methods. There are other pseudonyms for this, novel approach methodologies, etc. But generally, the term is approved as novel approach methods. So these are different approaches, novel approaches or new alternative methods that are aimed to replace the use of animal models in drug discovery. And this really has stemmed from things like the FDA Modernization Act 2.0 from back in 2022, and then more recently, the version 3.0 of this. And as you can see from the articles that I'm shown on the screen here, really the FDA is no longer requiring drug discovery efforts to have animal models used in them before entering clinical trials. Of course, animal models have been used historically as a surrogate for going from an in vitro cellular model into the complexity of a human being, effectively as a stepping stone to give confidence in going forward. But one of the problems is that they're not always that predictive. So a NAM could, in fact, be an in vitro platform, or it could be an in silico platform. As I'd mentioned, the advent of AI really fits into that sector. But ultimately, a NAM, to really fully replace an animal model or to replace it in a much more predictive manner, has to address the same factors of an in vivo model. And in an ideal world, of course, in a human system, because ultimately clinical testing takes place in a human being. So really, why are we looking at moving away from animal models, in vivo models? Unfortunately, we have seen over the years many examples of animal models that give either an indication that a drug will work in the clinic, and then it fails, maybe due to a lack of efficacy, or perhaps in vivo models that suggest a new drug is safe and tolerable. And yet when that drug gets into the clinic, it exhibits toxicity that wasn't seen in that animal model. So that then leads really to the subject of drug attrition. Unfortunately, using existing animal models, it really isn't working, and certainly not working as well as we would like. There are, of course, some cases where we have seen efficacy in animal models, and that is reflected in the human system. But like I say, it's not at a high enough level that we would really like to try and really reduce that attrition rate and improve the success rates of drugs when they enter clinical trials. An example of this is theralizumab, an anti-CD28 superagonist antibody that was being developed. When this was being developed, it passed all the necessary safety assays that were conducted at the time, and in fact, in GLP toxicity testing, showed no activity in non-human primates. So really, when you look back at the development of theralizumab, TeGenero, the company that were developing it, didn't do anything wrong. They did everything right at that time. Now, unfortunately, when theralizumab entered clinical trials, every one of the six subjects that were dosed with a suboptimal dose of theralizumab exhibited a cytokine storm-like result. And so I'm just summarizing that here, showing it in a simple in vitro study where we're looking at PBMCs, either human in the dark blue or non-human primate in the orange. And for the sake of clarity, I'm not including the positive and negative controls here. But what you can very easily see from this is looking at the non-human primate response, there's minimal to almost no response there. So that mirrors what was seen with theralizumab in preclinical development. However, when you look at the response in the human PBMCs, this is a very different story and indicates a significant cytokine storm-like effect, which unfortunately was seen in clinical testing for theralizumab. So this, of course, set in place the notion that really we should be testing for things like this, especially for biologics where they can potentially elicit a cytokine storm as part of the discovery process, where we are de-risking for the potential for this to occur in a human system. Another example here is a Pfizer compound that was in development, and this work has been published, the reference shown in the bottom left of the slide here. But this drug actually similarly passed safety assays and in fact showed no toxicity in GLP rat tox studies. However, when it got to testing in GLP non-human primate studies, a problem was seen. And so the question there is, can we do something about that? Are there ways that we can potentially predict that outside of an animal model and perhaps even in a human model? And I've showed some data here. We'll go into a little bit more detail on this in subsequent slides as we introduce some of the platforms that address the unmet needs that NAMs are really beginning to fill. So when we look at as I say here, the quest for more physiologically relevant solutions, what do we mean by this? Where have we come from? So we think about cellular assays, obviously an extension from having maybe done biophysical or biochemical assays as part of the discovery of a new drug, going into cellular models and showing that the drug works at the target in the context of a cell. Does it get into the cell if it needs to? Does it impact the target of that drug in a cellular context with the conformation of that target in a cell? We've then gone to things like mixed cell co-cultures, because in many cases, while we can still do use homogeneous cell culture, single cell types, they have their place. But co-cultures then begin to model the complexity of different cell types talking to each other. So right there, we've made a step towards something that is a little bit more physiologically relevant. This has come even further than that now, where we are seeing things that we know as organ on a chip. So mimics of different organs with appropriate cells, often in co-culture, sometimes in 3D as organoids. So running these on a chip where we can then evaluate the properties of new drugs in that more complex environment that is getting even closer to physiological relevance than perhaps even mixed cell co-cultures. Added into that then the engineering advancements within organ on a chip that includes microfluidics. So now not only can we look at the behavior of a drug in a complex cell system, we can look at the behavior of that drug under different circumstances. For example, mimicking PK with the fluctuating levels of the drug by co-administering with other drugs, either at different time points or with other agents that might be co-dosed with the drug. And again, beginning to try and mimic the complexity of the human body, where you have circulatory systems that ultimately will affect the efficacy and the distribution of a drug. And then ultimately, now as we're beginning to see more advanced examples there of whole body on a chip. So you can see the whole platform has really come a long way in terms of where we're looking and some of the models that we're really developing. And this really represents that quest for those more physiological solutions. And while most of the focus here is on human systems, this can also potentially be done in animal systems. But of course, we're trying to focus here on ways of replacing animal models in vitro, where we can conduct studies on human systems that are really predictive of what happens in the clinical scenario. If we think about some of the measurements that are made in these same scenarios, we think about earlier studies, we might look at simple biomarkers. It may be a simple single biomarker for a particular event. It may be a set of biomarkers. On the previous slide, I showed the release of several cytokines. There are technologies available to multiplex those evaluations. But things have come a lot further than that now. We are beginning to look, and in many cases, looking at panels of multiplex biomarkers, way beyond the half a dozen or so that I've showed before. So looking at many more biomarkers, often in context with each other, and to really begin to understand bigger changes, not just changes at the target or at a specific event, but more in a systemic or a whole body scenario. And again, extrapolating this to effectively the readout form of things like body on a chip, looking at biomarker networks If we perturb one biomarker within a complex system, what effect does that have on a variety of other biomarkers? It may be beneficial from an efficacy perspective, but of course it may also be damaging from a safety perspective. So these are all considerations that we have to think about when we're trying to identify novel alternative methods that get away from animal models. Ultimately, of course, with these larger platforms and the generation of massive amounts of data, there comes the potential for using AI or artificial intelligence. This has the ability to process and to munch huge amounts of data in relatively short amounts of time. Exercises that would typically take us as human beings a lot longer to execute. So this has the potential and ultimately the promise to really accelerate drug discovery and to really get us where we need to be faster with much larger data considerations, ultimately for improved success in clinical testing. Now, I'd mentioned earlier the quest for more physiologically relevant cellular models. So what I'm going to do is just introduce to you here a platform that we offer within Eurofins Discovery called the Biomap platform. And this is a good example where we have really taken advantage of mixing cells together in co-cultures and really trying to get a little bit more of a physiological understanding as to what a new test agent is doing. Biomap is a phenotypic translational cell biology platform, so it's an in vitro model, of course. But one of its strengths comes from the use of normal human primary cell types, as I say, often in mixed co-cultures together. So we can then establish models, or systems as we call them, where each system models a specific aspect of tissue biology or disease. Coupled with that is the use of particular stimuli in each system that accurately model that biology. So coupled with the cell types used, the co-cultures and the stimuli, of course, there come the readouts. And it's very important to make sure that the readouts one uses for a study like this are clinically relevant. So as we have put together biomap models, biomap systems, these have been extensively qualified by using and testing against them standard of care drugs and clinical entities where we know what the clinical response is. And thus the development of those models with the optimization that is used really in any assay development, not only with looking at the specific cell types that are used, the ratios of cells that are used, the stimuli that are used, but the responses of those biomarkers when that model is challenged with a drug for which we have clinical data. So we have then a platform that we can then use to profile new agents and evaluate what they're doing and how and why. Now, another aspect of the platform that makes this incredibly powerful is the database that is associated with it. We have tested over 4,500 agents in the Biomap platform, and this encompasses a huge range of standard of care drugs, approved drugs, clinical entities, things that have been into the clinic and failed because for various reasons they can provide very useful data that can help our understanding of new test agents in the context of this platform. We've tested a wide range of therapeutic modalities, small molecules, antibodies, peptides, recombinant therapeutic proteins, natural products, even mixtures in certain cases. So we have a vast diversity of mechanisms represented, target types represented, therapeutic modalities represented. And with that large database, when we profile an agent in Biomap, we generate what I always refer to as a phenotypic fingerprint. And we do that by measuring a large number of biomarkers. And I'll explain shortly how we do that. But it's that fingerprint that we can then go back to that database and we can say, does that fingerprint look like anything we've seen before? Or what does it have in common with certain drugs, certain test agents where we know what their properties are? So on the right-hand side of this slide, you can see with some of the quite sophisticated predictive analytical tools that we have available, we have the ability to probe various things. A lot of people are conducting phenotypic drug discovery at the current time. There may be a situation where a drug discoverer has a very desirable phenotypic endpoint which reflects modulation of the disease in their particular cellular platform, but they have no idea how their drug is actually doing that. So we can test that drug in Biomap, generate that fingerprint, go into our large database and say, what does it look like? For example, does this drug look like a JAK inhibitor? It's an immunomodulatory agent after all. And if we see suitable similarities there, and the similarities are all judged on a statistical basis, these are not subjective. So by assigning Pearson score correlations to these, we can determine if a similarity truly is significant or if it's a little bit more subjective or qualitative. Not only can we identify mechanisms of action or pathways that are impacted by a particular drug, either by design or outside of that, especially in the small molecule space, whereas we all know there are off-target activities that often can come into play, either in a good way or a bad way. We can look at potential toxicities, and we obviously appreciate that not everybody wants to go looking for toxicity, but because of the large number of drugs that we've profiled, we do have toxicity signatures that we have identified by looking at a vast range of drugs with different targets, different modalities, different mechanisms, but that share a common toxicity. And we've looked at those profiles and said, what particular biomarker changes are common to that diversity of drugs that share that toxicity? So we can take the profile of a new drug then and potentially look at it in context of the signatures that we have and say, does a particular drug have a potential toxicity liability? And right here, you can begin to see how this gets into that NAM space where we are predicting toxicities in a human system using data that is generated from a platform that has a very translational, clinically relevant foundation that hopefully will reduce attrition as those drugs go into clinical testing. We can, of course, look at things like combinations and the effects of two drugs together, not only on markers for efficacy, but also safety markers potentially. And we can, of course, benchmark. Ultimately, a particular drug will need to be evaluated in phase 3 trials if it's successful, and it will be compared against the standard of care drug there. We can do that. We can look at, for example, where there are differentiating features of the new drug that might suggest that ultimately, if it does show good efficacy and safety, it could have positive differentiation against the current standard of care for the indication for which it was developed. So we have the ability to have a platform at our disposal that is physiologically relevant. We've got a large database that can then support the application of this in the exploration and the profiling of new drugs through the advanced analytics that we have. So what does that look like when we put this into practice? This particular slide shows the example for a panel that we have, and typically where most people start in a Biomap study, called Diversity Plus. It comprises 12 different systems, 12 different models, each model reflecting a different aspect of tissue biology or disease. And really, just for the sake of illustration here, I'm showing 4 examples. In the blue rectangle, you can see 4 systems there that represent aspects of vascular biology. In the upside-down red T-shape, you see 4 systems that represent aspects of immune modulatory biology. So you can very quickly see there in the system where those 2 biologies overlap, that system is pretty good at looking at aspects of vascular inflammation. On the right-hand side, the upside-down yellow L shape looks at aspects of fibrosis. And then when you overlay that with the green upside-down L shape that looks at models that reflect aspects of respiratory biology, you can see 2 systems there, the MyoF and the BF4T system, that represent aspects of pulmonary fibrosis, either from a general perspective or a more focused perspective. So when profiling an agent across these 12 systems, we measure 148 clinically relevant protein biomarker endpoints across the whole panel. So we have more than just a small handful of biomarkers here, and this is what generates that fingerprint that I mentioned earlier. So if we then go to a Diversity Plus profile, this is the fingerprint to which I've been speaking. We have the 12 systems shown with the cartoons and the nominal labeling across the top. What I'd first like to draw your attention to is the dotted line and the gray signal. That represents the 95% significance envelope that is generated from historical data that looks at essentially the vehicle control. So just like any assay, there's a certain degree of variance that one sees in the measurement of each of the biomarkers. And the 148 biomarkers are stretched across the bottom there. I'm not showing them individually from the perspective of being a little clearer and not being too convoluted with the number of labels here. But that vehicle control essentially represents no change. We typically test over 4 concentrations, and those are shown in the red, orange, yellow, and green signals. And you can very nicely see here that when challenged with this particular test agent, there are changes in a number of biomarkers outside of that vehicle control. Just looking, for example, at the MyoF system, we see very few changes there. But looking at many of the other systems, for example, the CAS-M3C system, you can see there's some very nice dose-responsive changes here. So these are being manifested by the test agent that is being studied, where we see changes that occur over 2 or more consecutive concentrations at more than 20% outside of the vehicle control. And I should point out that the y-axis here is a log scale. We will annotate those. So in the BE3C system in the middle, you can see we've annotated IP10. On this particular trace, there would be many more annotated, but just for the sake of illustration here, I'm just showing IP10. We can also look at the effect of the test agent to see if it has an anti-proliferative effect or even a cytotoxic effect, as delineated by wide gray arrows or sharp black arrows, respectively. In this particular trace, we see the 2 systems, the SAG system and the BT system, where there is a degree of anti-proliferative activity seen. And this is important because it can potentially put some of those biomarker changes in context. Is the biomarker change mechanistic and related to modulation of what produces that biomarker, or is it related to modulation of the cell number? So you're modulating the biomarker ultimately by changing the number of cells that produce that biomarker. I'd mentioned a bit about toxicity signature analysis. So as I'd mentioned, when we look at a large number of drugs that share a common toxicity, and we look at their profiles, we can identify, and through data mining have identified key biomarker changes that are common to all of those drugs. The one thing they have in common being that they share a particular toxicity. So this leads to these toxicity signatures, and I have those outlined on the right-hand side here. For anybody interested, the reference at the bottom of the slide here goes into a lot more detail about this and actually shows you in a lot more detail how we've identified those toxicity signatures. But you can see for the different toxicities, there are certain drugs, and many of which we know, and we think, yes, well, we associate that drug with a particular toxicity that's well known. But don't forget, in this particular case, we're using the Biomap platform to try and predict, would we see toxicity in a human system, and really effectively circumventing the need for an animal model. As we discussed, they're not always predictive of toxicity, but can we get to an indication of toxicity earlier in drug discovery, where we can do something about it before the drug enters preclinical development and certainly before it enters clinical testing? Ultimately, that should help improve the success rate of drugs going into clinical trials and ultimately the benefit that we are providing to patients who are very much in need of newer medicines. If we look at a couple of examples here, I'm just showing the Biomap Diversity Plus trace at the top here, and this is for atorvastatin, which is an approved and prescribed HMG-CoA reductase inhibitor. You can see that there are various biomarker changes that are manifested by this drug. In certain cases, there are some anti-proliferative activities. Now, we know that atorvastatin is a tolerated drug. When we look at the toxicity signature analysis, and it is an in silico analysis of that fingerprint shown at the top of the slide, we see the summary in the table shown below. So there is immunosuppression. You can see that in particular in the BT system with that vast decrease in soluble IgG production. But there is also a degree of organ toxicity that is seen. And don't forget, these are over a range of concentrations that typically we will look at the Cmax and coming down to ultimately where one would expect therapeutic efficacy. So like any drug, this drug is not perfect. It does have a few toxicities that are associated with it. But when we look at one of the other statin drugs, in this particular case, rosuvastatin, this drug was also approved, but subsequently withdrawn. So when we look at this particular drug, you can see there are some similar responses. And admittedly, we've tested it here over 6 concentrations, so a couple of extra concentrations included here. But you can see, again, it shares the immunosuppression and organ toxicity that we saw for atorvastatin. But the interesting thing about rosuvastatin is that it was ultimately withdrawn due to the occurrence of severe rhabdomyolysis, which unfortunately in certain cases can result in fatal renal failure. So when we look at this profile in a little bit more detail, we do see modulation of biomarkers that are related to tissue homeostasis, in particular things like MMP1 and various others that are shown here. So it raises the question then, could potentially we have predicted the withdrawal of rosuvastatin here? So this is really going way beyond a novel alternative method for replacing an animal model, and raises the question, could we be at the dawn of being able to predict drug withdrawals before that drug is approved? And again, if done early enough, this could be something that could be addressed as part of that drug's optimization. I'd mentioned earlier the story around a Pfizer drug. This was an mGlu-R5 negative allosteric modulator that was being developed. And again, just in the spirit of full transparency, we can talk about this because this has been published. But Pfizer were developing this drug, and it showed no obvious toxicity in a variety of safety pharmacology assays, nor in rat models in GLP tox testing. The 14-day and 28-day rat tox models, it did not show any adverse effects. However, when tested in the GLP non-human primate studies at 14 days or 90 days, type 4 skin hypersensitivity was seen. So, of course, this raises the perplexing question that for such a clean, selective, and from the studies conducted, relatively safe molecule where we wouldn't be predicting anything in a non-human primate model, are there ways that we might be able to identify that? And could then a path be identified forward for that particular program to dispel or de-risk that particular drug? So this is the Biomap Diversity Plus profile of that Pfizer compound. And you can see for the most part, it's relatively inactive across many of the systems here. But there are some activities in the LPS, the SAG, and the BT system in particular. And what is particularly notable is the increase of soluble prostaglandin E2 in the LPS system and IL-2 and IL-6 in the BT system. Consequently, when you do the toxicity signature analysis of this, it's actually pretty clean across the board. And again, this is what we would expect based on the preclinical and even the rat clinic, non-clinical, the rat GLP toxicity studies. But what we see is one toxicity identified, and that toxicity was skin irritation at all 4 concentrations tested. So again, we're making the assumption here that in this particular human system, in this case, this may correlate with a non-human primate system. As I mentioned before, they don't always correlate, but in this particular case, what we were able to identify does make sense in the context of what was seen in that GLP monkey study. So the question then is, what could we do as a result of that to help that program? If you look at the biology and what results in production of the different agents here, IL-2, IL-6, and prostaglandin E2, that's summarized in the flow diagram on the left-hand side, and ultimately how this can result in skin irritation on the right-hand side. So one could conceive a particular scenario here where you could use these data and then put in place simple assays and with a medicinal chemistry campaign, dial these unwanted profiles out of the molecule's overall profile. Ultimately, this should then reduce or eliminate that ability or unwanted activity to cause skin irritation. If we think about other aspects of the biomap platform, again, really capitalizing on the use of mixed co-cultures of cells, this is an example where we're looking at reconstitution of the tumor microenvironment from an oncology perspective. We're looking at mixing either fibroblasts, immune cells, and a tumor cell, in this particular case HT29, a colorectal cancer cell line, to represent the stromal matrix, or mixing together the cancer cell line, PBMCs, and vascular umbilical vein endothelial cells to model the vascular component of the tumor microenvironment. What you can see here is when we evaluate the effect of, in this particular case, anti-PD-1 or anti-PD-L1 drugs, we see this restoration of immune response. This is, in fact, how these drugs work. They're all immuno-oncology agents, durvalumab, nivolumab, and pembrolizumab. And what you see here is this nice reflection of what is seen clinically. So when we look at how these drugs work, people sometimes are mistakenly under the impression that they actually activate the immune system. They don't. They restore the ability of the immune system to respond in the presence of the tumor cells and within the context of the tumor. I'm not showing the data here, but when in fact we look at these drugs in the Diversity Plus platform, the broader biology, what you might associate more with the systemic exposure, we actually see very little activity at all. We certainly don't see T-cell activation. So this shows that what we're seeing here models very nicely what is seen in tumors when treated with these checkpoint inhibitors, and that reflects what is seen clinically and is why they work so well. It also raises a very interesting question that if we look at other drugs, and I'm showing here 3 drugs that were around long before the idea of immuno-oncology: erlotinib, a targeted EGF receptor tyrosine kinase inhibitor, gemcitabine, and paclitaxel. When you look at these drugs in the same system, you can see also some degree of immuno-oncology potential with them. Now, obviously, they have efficacy through other mechanisms, whether it's kinase inhibition, whether it's acting as a nucleoside analog, or modulating tubulin dynamics. It could well be that these particular qualities, these immuno-oncology qualities that are seen here, contribute to the overall efficacy of those drugs, which is why they've been so successful. So really, we can see here that how these drugs are working, and we can actually explain, in this particular case, the whole concept and demonstrate this of immuno-oncology in an in vitro system. And again, without the need for an animal model. So we're now going to change gears a little bit and talk about how we can get the most out of existing platforms. As I'd mentioned at the outset, we have a lot of platforms that have been in existence for many years now. And how can we really begin to think about employing those as novel alternative methods? What I'm going to talk about here is the field of cardiotoxicity. So really looking around some of the modulation of ion channels, which has been central to evaluating and de-risking for cardiac toxicity for a great many years now. When we look at a variety of ion channels that are involved in cardiac safety, the hERG channel, the human etheragogo channel, which is a potassium channel, immediately comes to mind. But there are actually a number of other channels that work in concert with hERG, and ultimately these are all responsible for maintaining the appropriate dynamics of the heartbeat from the potential of electrophysiology. So if we think about evaluating these ion channels together rather than in isolation, this has led to the generation of what is called the Comprehensive In Vitro Proarrhythmia Assay, or CIPA for short. If we look at the history behind this, as I mentioned, hERG testing has long been a gold standard in drug discovery. When you develop a molecule, in particular small molecules, the sort of point that was made there was that one shouldn't inhibit hERG. So when we look at this, there have been a lot of drugs that have been withdrawn due to hERG blockade, and they're shown on the right here and the years in which they were withdrawn. But what flies in the face of this then really is that there are some drugs that do inhibit hERG but they're actually quite safe. So really what we need to be able to put in place is a platform whereby we can evaluate for the risks, the QT prolongation risk that is associated with hERG inhibition, but where we can screen for drugs that, although they may inhibit hERG, they may inhibit other ion channels that negate that effect. So the concurrent inhibition of different ion channels really does need to be considered here. So this has really led to a new way of thinking for how we explore cardiac safety evaluation. So just going beyond hERG, as I'd mentioned, inhibition of hERG alone results in QT prolongation, which can lead to potentially fatal proarrhythmias. However, hERG inhibition, fundamentally just hERG inhibition, doesn't necessarily equate to that. If we look at something like verapamil, it's an approved antiarrhythmic drug, ironically. It blocks hERG, but it also inhibits the CAV 1.2, the voltage-gated calcium 1.2 channel, and it's quite safe, at least in the context of its therapeutic use. If we look at ranolazine, as another approved drug, it also blocks hERG, but it inhibits the sodium late current channel, and again, it is also safe for use. So this very nicely demonstrates the need for this new paradigm where we're looking at the comprehensive in vitro proarrhythmia assay. So looking at these ion channels together. And this has really reflected a change in thinking as to how cardiac safety has been addressed. And again, gets to using current platforms as a way to improve the chances of clinical success, where we really don't have to rely on seeing cardiac issues within an animal model. So if we think about this and some of the things that have come out of the CIPA consortium, we're very proud to be members of the development of the CIPA assay, or the CIPA assay platform, as a result of being part of this consortium. It's really led to the harmonization of the way in which we conduct ion channel assays. It's allowed regulators to look at cardiac safety studies a lot more quickly than they've been able to do previously, and across a variety of different submissions using harmonized protocols. Another aspect of this is that it integrates both risk assessment from a non-clinical perspective using non-clinical data and clinical data. What we see here is reflected by clinical observations with some of those drugs that have been evaluated previously. It does provide a totality of evidence approach, and this is becoming more of a growing theme in various aspects of drug discovery, not just here within cardiac safety, but also in things like looking for and de-risking for carcinogenicity. We're also able to inform really on late phase clinical trial design and labeling for market application. And again, understanding the potential for any of the drugs going forward for cardiac safety issues. They may be tolerable safety issues, but it does inform on this much earlier. It's also reduced, as I've got here, the number of thorough QT or QTC clinical studies that are needed. So overall, it's reducing the complexity of clinical testing and, of course, the preclinical side of that with animal testing to really accelerate and make the prediction of cardiac safety a lot more reliable. And ultimately, this, of course, has resulted in a tremendous reduction in cost, and perhaps most importantly, from the perspective of the patients, improving safety. If we look at really where things have come, going back as far as before the 1990s, like I say, hERG was identified as being a key player in potential cardiac problems. But you can see here from the timeline that there have been advances that have been made and are steady in the field. And these have actually resulted in changes in the ways in which regulatory guidelines are set out. And one of those more recent examples here, as I'm showing, is the evaluation of drugs for cardiac safety in vitro models at 37 degrees C, so at physiological temperature. So I'm going to show you some data that really outline that in a couple of slides' time. But ultimately, like I say, this has really impacted the way in which regulatory guidelines are set forth. It's changed the way we think about cardiac safety evaluation from the in vitro perspective. And really now we are at the point where we can do a much better job of de-risking for this. And as I'd mentioned, this has reduced the risk on more exotic clinical studies, as well as some of those animal in vivo studies that have been used historically. Just a quick snapshot here to show some data supporting the idea of testing at 37 degrees C versus room temperature. You can see that for many different drugs, and some of them are exemplified here, going from room temperature testing in the blue, we're often seeing a change in the apparent or measured potency at 37 degrees C at physiological temperature. That's shown in the green traces here. And you can see, for example, for sotalol at the bottom in the middle, that change is quite significant. So this is just one of many advances that have been made in this particular field. We're very proud members of the HESI organization here at Eurofins Discovery. And ultimately, this is still pushing in all aspects of moving the field forward for working towards a better way of cardiovascular safety assessment. And in fact, just as you'll note at the bottom here, the FDA Modernization Act 2.0 really authorizes the use of certain alternatives to animal testing, and that includes cell-based assays such as these, as well as computer models, to really get an exemption by the US FDA at least that allows the investigation of safety and effectiveness of a new drug. What we're going to do now is take a very quick look at aspects of safety pharmacology. So safety, of course, outside of cardiac safety has to be evaluated. And there are many different ways of looking at that, both within and outside of cardiac safety. I'm just using cardiac safety here as an example. We have a number of different assays. Many assays have been around for quite some time. In fact, back in 2012, a seminal paper by Bose et al. That was the result of a consortium of pharma companies that got together and identified a variety of targets that are key bad players in the safety pharmacology field. And that really shaped the putting in place of safety pharmacology panels. And we offer those panels and have been doing so for many years now. But when we look at these various assays, they can all reflect different parts of physiology, whether it's contractility of heart cells, whether it's looking at things like heart rate and blood pressure, or even structural aspects of the coronary tissue. The assays can relate to genetic, pharmacologic, and clinical adverse events, the basis by which they're put in place. We have binding assays. We have looking at ligand displacement, but we also have enzymatic or mechanistic assays, and some of these are kinase assays that are shown here. The little table at the bottom gives a breakdown by different target types, and some of them represent things like calcium flux in iPSC-derived cardiomyocytes. So these assays are very good at distinguishing things by mechanism of action, and they have a very good place. Like I say, they're not the be-all and end-all of drug discovery from a safety perspective, but they do give a very good idea of what potentially could be a liability early on. And they are, as a result, used routinely for screening. So these assays have a very firm and a very good place in drug discovery. Now, just this last year, the consortium, the IQ-DRUSAFE consortium, got together and again involved many different pharma companies that came together and re-reviewed the safety panels, and they updated them by the inclusion of other targets. And our panels now go some way to actually reflect that. We've included those new targets in our panels, so that then improves the ability of those panels to de-risk and dispel safety, potential safety liabilities earlier in drug discovery. As I show in the flow at the top there, we can use these at really any stage from hit finding right the way through to candidate selection. Really the only difference being the complexity of the panel that is used and the degree of profiling that is done or needed in certain cases. So much so that we actually have obviously a variety of safety pharmacology assays at our disposal. And most recently, we've actually employed the ability to present these data graphically, and just shown in the little chart at the bottom left there, this shows the potential for different safety liabilities and looking at different doses. And in fact, we can even associate that now. We have an offering that we provide where we look at the correlation of that with known toxicities, where those particular safety pharmacology targets are particularly linked with a certain toxicity. And I alluded to aspects of that on the previous slide. So again, these are panels that have been in place for quite some years now, but the constant optimization of them is helping to reduce those failures, either the need for doing it in essentially unpredictive animal models, but the reduction of potential failures in phase 1 trials. So it's all working together. No one platform is working alone here. I'd mentioned at the outset, of course, the advent of artificial intelligence, AI. We're all familiar with this, and we've all seen it, I'm sure, in different areas of drug discovery. And artificial intelligence really is beginning to show that it has great potential in different areas, looking at improving efficacy, for example, looking at improving safety, looking at improving ADME and PK qualities. But what is artificial intelligence? What are the different kinds that are available? Well, there is agentic artificial intelligence, but there's also another form called generative AI. So these two differ in the way in which they process and they provide potential solutions. So agentic AI will actually act autonomously, will act on its own, and it will then provide as a readout workflows, screening paradigms, things like this. It will automate workflows, which, as you can probably pretty quickly see, is important for drug discovery. What is the most logical workflow that we need for a particular program? Generative AI is actually the area of AI that we tend to most commonly associate with drug discovery. This is where the output of the AI platform is based on the input. You provide the AI platform with a tranche of data that addresses a particular parameter. The platform will evaluate it and then make predictions based on the data. And the more data you fuel that AI engine with, the better the prediction will be. And we tend to term this things like machine learning. So that output, what we do is we use it to create novel molecules or better understanding of pathways. So you can very quickly see that both the agentic and the generative aspects of AI are very important for drug discovery and essentially can be used together. Now, when we put that in context of what is done for drug discovery, I've summarized that at the bottom here. When you look at the number of potential molecular compounds that could exist, you're looking at a huge number here, 10 to the power 60. It's not just a large number, it's not a very large number, it's an absolutely huge number. And I haven't done the back of the envelope calculations on this, but if you were to be screening billions of compounds per day, you could be looking at billions of years or longer to be able to screen through all of these. Obviously, that's not a practical solution. So if we can use AI, we can screen through huge numbers of compounds based on input with manageable numbers for which we can generate data. So we can import the data for those molecules, we can import the structure, we can import the data coming from a particular assay. It may be a safety pharmacology assay. It may be a protein binding assay for ADME. It may be biomap data. It can be any form of data. We can then get predictions from that generative AI platform. We can rank order those compounds that are proposed by that output. And we can say, well, these compounds look to be the most promising. And so we can then confirm that by assay. And so, in fact, here at Eurofins Discovery, we do a lot of work with a number of people in the AI area where we can either provide data that can help fuel and can help validate and qualify AI engines, and at the back end, provide those confirmatory assays that then help qualify that engine even further and say, yes, these compounds, these molecules that were proposed by the engine look to do exactly what that engine was designed to predict. And the more assay data like that you can feed back into the engine, the better that engine becomes at predicting the molecule's properties. So I think we're really at the dawn of something exciting here, where we have a lot of data at our fingertips that we can use in these models. And like I say, this is something that has really gripped the drug discovery field and taken it by storm over the last few years. So one last thought here, and like I said, this is not a comprehensive view of every aspect of NAMs, but has given a flavor, hopefully, of some of the things that we can do and are doing to get around the historical reliance on unpredictable animal models. But one last question I'd like to leave you with is, do we really already have a wealth of data available that effectively is NAM data? Well, the answer to that potentially is yes. We've got a lot of data that has come from the historical conducting of clinical trials. This is called real-world data. So if you think about it, coming back to some of the premises that I set out at the start of this webinar, these are data that have come from a human in vivo system. Effectively, it's the perfect NAM. So we have those data, so why wouldn't we be able to use those data, maybe in concert with AI platforms, AI algorithms, to generate much safer and efficacious drugs going forward? So with that, I'd like to just summarize some of the things that we've talked about in the pursuit of NAMs and why they're important in drug discovery. I like this statement, this quote from Lily Tomlin: "The road to success is always under construction." I think you can see that quite easily with things like the herd testing for cardiac toxicity and the safety panels, but also the evolution of cellular models, the co-culture models, biomap, organ on a chip, and things like that. We've historically relied on animal models, but that historical reliance is rapidly changing. The FDA Modernization Act 2.0 has really crystallized that, but many other organizations around the world are embracing this and taking this forward, things like the NC3Rs and things like this. Really, the key to this is to having models that are more predictive, but are human relevant. Everything I've spoken about from the in vitro cellular perspective in this webinar has been a human platform. And ultimately, that's where we are using these drugs. Now, of course, there are adaptations of some of the drugs where we can use those in veterinary scenarios. But in those particular cases, you could put in place models that might model that based around data that we have in certain species. But ultimately, it's these better platforms that are needed across the industry to help replace that reliance on animal models. As we've seen, they can encompass new technologies and approaches. I think nothing really exemplifies that better than the AI area right now. But some of those cellular models, those advanced, complex cellular models, really help illustrate that very nicely. As I mentioned, though, we have a lot of platforms already available, and they continue to add value, but with refinements. And I think we've seen that quite nicely with the cardiac safety panels and the hERG and ion channel testing there. And we do have a lot of data available already, not just in the clinical quarter, but also in company repositories, in even Eurofin's data repositories from the historic testing of lots of different agents. We have tested lots of approved drugs, standard of care agents, as I mentioned in the biomap platform. We've looked at lots of things that we can procure commercially. They're in the public domain, and tested them against a vast wealth of different assays. So we alone have a lot of data, but there are many other organizations out there that also have data that can all help fuel these approaches. And that really is why we are happy to be members of the Pistoia Alliance, the HESI Consortium, and things like that, sharing the data that we have and then working with others to help really move this field forward. like I say, AI is really changing the whole way that we think about drug discovery, and I think there is great potential here. We have yet to really fully realize that potential, but I think over the coming years we will see that. And in hand in harness with these other approaches, I think we will go a long way. So really, the era of novel approach methods, or NAMs, is now. We are at the stage where we are really embracing the challenge and using what we have available and the new technologies to try and push this forward. So I hope that has been a very informative webinar for you. I'd like to thank you all very much for your time and attention, and I would be very happy to answer any questions you may have.
Wow. Thank you so much, Alistair. What an excellent and informative presentation. And I just want to remind the audience that you can ask questions through the Q&A box on your screen. And with that, we will open it up to our first audience question. Alistair, this audience member is asking, what specific type of assay or platform qualifies for use as a NAM?
Yes, thank you. That's an excellent question. Really, any kind of assay platform or technological approach could potentially be used as a NAM. The key is that it has to model human physiological response accurately and in a way that we see when we test a drug in a clinical scenario. There are plenty of assays, and I think some of the ones that we just saw in the webinar that have been around for a while, but they can serve the role as a NAM, and especially as we refine and advance those technologies, we add more sophistication to them, and that sophistication is really pushing them towards physiological relevance. So I think really any model, and it can be, as I think I mentioned at the outset, either a laboratory type model or an AI approach, as long as it models the clinical outcome accurately, then really anything can be used as a NAM there.
Okay, thank you. This audience member is asking, how does the biomap platform address the possibility of donor-to-donor variation with the primary cells used?
Yes, that's a good question. We do make extensive use of normal human primary cell types in the biomap platform, and we are obviously aware of the potential for donor-to-donor variation. The way we get around that is we actually use pools of cells. So for any particular cell type, we will pool together several donors, typically 3 to 6 donors. And what we do is we qualify that pool so we know how each individual cell type should behave when presented with various controls, various reference controls. So when we pool a number of donors together, we will evaluate them with a certain set of controls, and if they pass QC on that, then they are used together in the various models or the systems, and those models or systems have their own controls. So there are multiple levels of QC effectively there. Sometimes people will ask from the immune cell perspective, what about the potential for mixed lymphocyte reaction? Really, none of the exposures that we use, the incubation times, are long enough to manifest a mixed lymphocyte reaction. So that actually is not really a problem that we encounter within the context of these studies.
Okay, thank you so much. This audience member is asking, how is it that existing technology platforms, like the cardiotoxicity ion channel one that you mentioned, can be used as a NAM?
Yes, so again, it's really making that link between a traditional in vitro type assay. The ion channel approaches have been around for many years, but it's really the moving of the way in which those studies are executed to something that is more physiological. One of the parameters that I mentioned was, in fact, now conducting those studies at 37 degrees C, or physiological temperature. So that's one of several steps that have been made by the community in trying to get those assays more physiologically relevant. So again, it comes back to the premise that the closer an assay or a technology or even an in silico platform can be in terms of its output to what is actually seen clinically, the better it is and the more like a NAM it becomes. And really, I think the qualification of those platforms using drugs where we know what the clinical outcome is and making sure that what we see in vitro mirrors that clinical outcome exactly, that really is the key to progressing those to becoming true NAM type technologies.
Thank you. This audience member is asking, what are the drawbacks of current NAM approaches and how should they be resolved?
Yeah, that's a good question. There are, with any kind of assay platform, there are always caveats. We can build advantages into them, like I say, with physiological relevance. Just taking the Biomap platform, for example, one of the limitations there, it's not in the same category as things like organ on a chip, even though the physiological context is there. With the current Biomap platform as we have it, we can't model the fluctuating level of drug that's exposed in a PK sense, a pharmacokinetic sense. So in terms of the limitations, or the drawbacks of some of the current NAM approaches, I think it lies really in their sophistication. I think we've seen with things like the ion channel type approaches for cardiotoxicity, they started out fairly simply, and then we've expanded those and we've made them more sophisticated. There are always improvements that can be made. I think the quote that I had, that the road to success is always under construction, rings true here, and there are always improvements that can be made, really, with any of the platforms, and that includes AI platforms. One of the key things for an AI platform is that you feed it good data to start with, and then you gradually improve the data, and it's a cyclic kind of thing that you generate then molecules that can be tested, and then you generate data from that and you put that back into the model. So even there, the AI models are good examples of where through machine learning they are becoming better and better and better.
Okay, thank you very much. This audience member is asking, how do the applications of NAMs for efficacy compare to safety assessments? Great question.
So the various NAM approaches that we have with safety, I think we've seen fairly well with something like the Biomap platform, where we've qualified the platform for toxicity signatures and various safety liabilities there. And we've qualified the platform by extensive testing with known drugs, for example, drugs that have the same kind of toxicity. And so when you've done that kind of qualification, and it's not simple, you have to do it with quite a number of test agents to make sure that the reproducibility of the system in predicting a clinical outcome is real, then that gets you to being in a position where your model can more accurately predict what the expected or the ultimately seen safety perspective is actually manifested clinically.
Okay, thank you so much. This audience member is asking, by using an alternative model like AI models instead of animal models, if a lead compound passes the test, then we go to the next step on a clinical trial of that lead compound on humans, or we use an animal model before a human trial.
Yeah, that's an interesting question.
Yeah.
So one of the key things about NAMs is that they are emerging and they're being put in place because many of the animal models just aren't predictive. So it would seem retrograde to come to a conclusion or a potential solution using a NAM and then to go back to an animal model to effectively test or corroborate the conclusions that you've obtained from the NAM. What I would say, in certain cases, though, pharmacokinetic models can potentially be still used from a NAM perspective. But when you get into the complex, the really complex biology of efficacy and even some of the safety profiles, that's where I think going back into an animal model is probably not an advisable step. We've seen certainly over the years that there are animal models that do show efficacy and do in certain cases predict potential safety issues. But I think with some of the examples that we discussed in the webinar, that's not always the case. And unfortunately, in certain cases, it can be pretty catastrophic. So my personal feeling is that going back to an animal model is not necessarily advisable. I think if the NAM itself, the technology or the platform itself, is thoroughly and fully qualified, and like I say, qualified with drugs where we know what the clinical outcome is, such that the NAM itself also demonstrates that same clinical outcome, then really there is no need to go back to that animal model for confirmation.
Thank you so much. This audience member is asking, are there any specific methodologies which can be used with AI or ML?
Yeah, really quite a lot of different methodologies. We're on the cusp of a very interesting and very exciting era in drug discovery, and the potential, I think, of AI, I think we're really just scratching at the surface. There are lots of AI engines and approaches that are looking at a variety of different aspects of drug discovery. For example, safety pharmacology, ADME, tox properties, and things like that, and even in certain cases, efficacy. Simply put, if you can take data, and again, as we'd said earlier, if those data are robust, reliable, and truly predictive of what you would ultimately see in a clinical setting, then really any of those could be used in an AI context. What I would love to see, and I think we're a few years off this just yet, is where an AI model can actually take all of those different approaches, as we would in a conventional drug discovery program, and evaluate them together. For example, do you need to get a certain amount of drug exposure to get efficacy? And with the drug that the AI platform is predicting, do those two align? Can you get that predicted exposure? So effectively going from I suppose what you might consider sort of a univariate approach to a multivariate approach, that to me, I think, is where the true power lies. How far off we are from that, I couldn't say, but I think when we are at that stage, that really will be the kind of scenario where we can put in all the different parameters that, like I say, in a traditional drug discovery program, we evaluate systematically, and you put those into an AI platform and it can say, here is the drug for this particular disease. And when we think about things in the current thinking in drug discovery, especially in a world of tailored or personalized medicine, one could envisage really the scenario where your AI platform will take the individual person and identify a drug for them. That would be really exciting, I think, when we're at that stage.
Thank you so much. This audience member is asking, how good is your platform in predicting potential off-target toxicities due to the manifestation of long-term drug exposure in chronic administration scenario?
Yeah, that's actually a very interesting question. So with the Biomap platform, as I mentioned, we have identified a number of toxicity signatures. And just to recap on that, the way we did that was we looked at a wide selection of drugs of different target types, different modalities, but they share a common toxicity. So when you then look at those phenotypic fingerprints and you compare them, you can identify specific biomarker profiles or signatures that reflect that toxicity. One of the things that we have actually done, and actually presented this in toxicology meetings, is exactly that question. Could we potentially predict drug withdrawals and attrition that way? And we've actually looked at a couple of examples where when we look closely at the Biomap profile, between two drugs that are very similar and target the same molecular mechanism, both are approved, but then one is subsequently withdrawn. In certain cases, potentially we are able to identify markers there that raises the question, is that an early indicator that with long-term exposure to that drug, that would ultimately lead to the problems that were seen with that particular drug and ultimately resulted in its withdrawal. So I would say, in answer to the question, potentially yes. I think we need to conduct a lot more case studies of that kind to be able to say with certainty that we can do that. But I think with what we've done initially, we would say, yes, there is the potential we can do that.
Okay. We have about time. We're going to squeak in one more question. This one is asking, can biomap assays be used to identify PK and in vivo stability risks? Can these assays replace animal testing for pharmacokinetics?
Yeah, that's a good question. On the surface, I would say no.
Okay.
But what we can do is we can profile at different concentrations. So it's not quite the same where you're exposing to a drug and then gradually tapering the drug off. And that's where microfluidics would really come into their own. But we can do it by looking at different concentrations and then saying, you know, at a Cmax concentration, we might see this effect. And in fact, when we conduct diversity plus type studies, the kinds that I discussed in the webinar, a common question that we get asked is, what concentration range should we examine? And I would always suggest going a little bit above the anticipated Cmax and then coming back down ultimately to where you would expect to see therapeutic efficacy, because that defines your therapeutic window. Can biomap itself predict the PK exposure? Not really. It's not really set up to do that. It's set up to look at other things. Of course, we do have other approaches to be able to look at PK, and certainly Eurofins offers a wide spectrum of ADME related activities, both from the in vitro perspective, as well as looking at in vivo PK, admittedly, again, with animal models there, but being able to conduct a typical 4-box analysis where you can predict human exposure. That, I think, those kinds of animal models, I think, will be around for quite a while.
Okay. That's all the time that we have. Thank you again, Dr. Alistair King, for that great and, again, extremely informative presentation.
Thank you. My very great pleasure.
Excellent. Any questions that we didn't get to during the webinar will be answered by email. Thank you, participants, for being a great audience, and On24 for the technology and production services. A big thank you to Eurofins Discovery for the sponsorship that made this interactive webcast possible. Please be sure to check CNN or CNN Online for information on the next edition of CNN webinars. And for CNN webinars, I'm Melissa O'Mara. Goodbye.